Did you know that we can use lasers to control brain activity, and that same technology is being used to restore certain elements of vision to blind patients? If you're intrigued, stay with me, because my entire PhD was on doing optogenetics the right way. So starting with brain cells, they're not naturally responsive to laser light, right? I mean, maybe if you heat up the brain enough with the laser, you're going to start to change its activity, but at that point you have a whole set of other problems. So first we've got to do some genetic engineering to make those cells sensitive to light, and then we can start turning different parts of the animal brain on and off to see what they do. And that's going to help us as humans learn more about ourselves, because humans and animals are actually really similar, both genetically and in terms of our larger, more visible structures. So for example, optogenetics has been used to investigate memory formation, and one of my all-time favorite papers is on this topic. But we can also use it to investigate the ways in which things go wrong during disease. So that's a big one, right? For my PhD work, I was using optogenetics to try and figure out where and how the feel-good effects of opiate drugs are processed in the brain. So what are the brain chemicals that are involved? What are the electrical activity patterns that are involved? Do those activity patterns actually matter, or can the brain just function in this active or inactive binary sort of state of information encoding? I'm actually still involved in this work to this day. I love it. I think it's so cool. Now, believe it or not, this is just the tip of the iceberg when it comes to optogenetic tools. It can do more than just help us push buttons in the brain and see what those parts do, even though that's pretty impactful. Cell biologists can use it to control specific molecular interactions within a cell using light to try and understand them better. Vision scientists are using it to try and make the eyes sensitive to light again. In fact, there was just a clinical trial on this very topic, vision restoration using optogenetics, that reported positive results this past March 2025. But how does it work? The quick version is that we can genetically engineer cells so that they respond to different wavelengths of light. So yellow light, red light, blue light, that sort of thing. Sometimes we can even get them to respond to more than one color at a time, and that's useful in the vision field, for example. Now, the nature of that cell's response depends on the cell type and what it is that you're actually studying. In its most widely used forms, we'll work with a gene that codes for a light-sensitive protein. That protein will either be a channel or a pump for ions like sodium or chloride. So ultimately what this protein will do is affect the electrical properties of the cell. And this protein is called an opsin. We can find these opsins in microorganisms or even insects. So we get them from bacteria, algae, even mosquitoes. We then deliver these opsin genes to the cells that we want to study. So it's either in a dish or into the animal itself. Now, in the case of neurons, activation of an opsin reproduces the electrochemical phenomenon that leads to an electrical impulse in the brain, and that's arguably the basic unit of information in the brain at the cellular level. Now, this is relatively easy to do in animals. It's almost become routine, but when it comes to humans, there are still a few challenges that we have to overcome. Why would we even want to do this in humans? Well, you have diseases like Parkinson's disease or epilepsy that could actually benefit from optogenetics as a potential future treatment. A few examples of challenges when it comes to humans. Our brains are just much bigger than rodent brains that we usually work with, and so you need to get a lot more light in. That can be difficult. Safe delivery of the gene is another big one, but as the technology continues to improve, I think it's genuinely reason for optimism in this field. We're not quite there yet for the human brain, but for human vision, I mean, that's in clinical trials right now. Of course, some of you might be wondering whether or not it's possible for someone to use optogenetics on you to control your brain activity without your knowledge. And I'm just going to tell you, not possible, okay? Unless you're walking around with implants in your brain right now, having previously had injections into your brain to do the genetic engineering that's required to make it work, don't worry about it. And by the way, what I mean by implant is a fiber optic cable attached to an LED, so like the actual light source itself. Nobody's playing with your brain using opto, all right? You can breathe easy. You're now up to date on optogenetics. If you learned something, share this video with a friend.